stimulus-specific plasticity in human visual gamma band activity. So one finding that has been reported in a lot of different model systems and a lot of different sensory areas is that if an input stimulus is repeated, we usually see a decreased and a sparse neuronal response. So for example, this has been measured in area V1 firing rates and area IT firing rates in the response to visual stimuli, as you can see on the right. And at the same time, we see stable or increased perceptual performance. So this raises the interesting question, how does the brain do more with less? And one phenomenon that has been connected to this is that if a stimulus is repeated, we usually also see an increased amplitude of stimulus-induced gamma oscillations. So you can see on the right gamma oscillations that have been recorded in macaque area V1 that increase in strength as a grating stimulus is repeated. So the aims of this study was to test if this gamma repetition enhancement effect is A, specific to the stimulus that is being shown, or if it's just an effect of running a visual experiment, and B, if it is persistent in time and de-adaptation, which would be a requirement for it to be some sort of learning mechanism. So we recorded 30 human participants in MEG. We showed them grating stimuli. There was a simple change detection task. And we showed 600 gratings in total. And for 120 trials, we would show the first grating, grating A. We would switch the grating and show 120 trials of another grating, switch again. And then at the end, we would show 120 presentations of the first grating again. And between the first and the last block, there was about 25 minutes of other gratings. And there was no break or anything between the blocks. We see a strong gamma band response to these stimuli in the early visual cortex. So this is source localized MEG. You can see on the left a spectrum of the MEG signal that has been source localized. You can see a clear gamma band response between 40 and 60 hertz. You can see on the right the same spectrum plotted over time. You can see that this is a banded response. If we localize this response over the brain, we can see that it is strongest in areas V1 to V4. And we can take the gamma band strength that we get for each participant and for each trial. Can extract this from our data, and we can plot this over our 600 trials, over our grating repetitions. If we do that, we get the following plot. So when we show a grating for the first time, as you can see on the left, we get a strong response, then drops down, and then over the following 120 repetitions, we see an increase. If we then switch the grating, as you can see in the color switch, we see the same pattern again. We switch the grating, we see it again. We switch the grating, we see it again. If at the end we repeat the same grating from the first block, you can see that there's the same pattern, but it starts from a higher offset. You can basically subtract the last block from the first block as a rough estimate, and there are also more complex analyses on this in the paper. We see a similar pattern in the frequency of the induced gamma band response. We don't see this initial decrease, but we see an increase over the repetitions of the different gratings. And we can see that this effect is specific to the gamma band. So here we're plotting the spectrum from earlier for 20 trial bins of the repetitions. You can see that the increase is specific within 40 to 60 hertz. If we want to find out where this effect happens in the brain, we can basically fit a line to the repetitions of each stimulus, excluding the first few repetitions. And we can do this for each position in the brain. And the resulting plot is shown here, so we can see that we see the strongest increases in early visual areas. Now, you might notice that in this pattern, there's a really strong response on the first trial. This wasn't something we expected from earlier data. And we asked ourselves, where does this early gamma decrease come from? And one idea we had was that it might be related to an arousal response the participants get once a new grating is shown. So we looked at the pupil size at stimulus onset. You can see that when the stimulus comes on, pupil size decreases because the stimulus is a bit brighter than the background. But this pupil constriction gets smaller with stimulus repetitions. And if we just overlay the amount of pupil constriction we see on each stimulus repetition to the induced gamma change, we can see that they show a very similar pattern for the first few trials, but then pupil constriction stays flat while gamma power increases. So this leads us to assume that maybe there's something arousal related going on during the first few trials. Something we also wanted to look at was frequency resolved Granger causality. So basically the ability to predict signals from higher brain areas from the signals in lower brain areas and vice versa. So what I'm plotting here is Granger causality between area V1 and V4, in green in the feed-forward direction, in black in the feedback direction, and in the lighter colors during the later repetitions. And you can see that there's this gamma bump in the feed-forward Granger causality between V1 and V4, and that this increases with stimulus repetitions. We can look at all brain areas, and I'm plotting in green again the feed-forward pairs, in gray the feedback pairs, and just look at where do these changes happen, and you can see that they happen exclusively in the gamma band and most strongly in the feed-forward direction. And here we're just taking the strength of the changes in the feed-forward and the feedback direction, and we can see that the feed-forward Granger causality increases in strength. So to sum up, stimulus repetition increases gamma power and frequency. This effect is stimulus specific. It stays around over 25 minutes, even when we're showing different gratings. We figure that the initial gamma power decrease might well be arousal driven, and we also see an increase in feed-forward gamma functional connectivity with repetitions. You can find further analyses and a lot of things I didn't talk about in the full study, which is in the description. And I'd like to thank you for coming to my Flash Talk and wish you a nice remaining conference.